Hi, everyone. My name is Abby Ryan, and I'm part of the marketing team here at Austin Pharmaceutics. Today, Dave Miller, our Chief Scientific Officer, will walk you through an overview of Austin Pharmaceutics, our formulation and manufacturing capabilities, and KinetiSol, our next generation amorphous solid dispersion technology. He'll also share how our team helps sponsors navigate complex oral drug development challenges and identify a clear path forward. So with that, Dave, I'll go ahead and turn it over to you.
Great. Thank you, Abby, and thank you, everyone. Welcome. Thank you for taking the time to learn more about AustinPx. As Abby said, I'm Dave Miller, the Chief Scientific Officer and one of the co-inventors of our KinetiSol technology. So over the next 30 minutes, I'll walk you through who we are, our science and technology, and our development and manufacturing capabilities, and supplement that with a few real client case studies from our archives. So first, let's talk about complexity in our industry. So modern drug development is shaped by a lot of forces that extend beyond the molecule itself. We've seen a real surge of innovation with complex molecules and molecules whose properties go beyond, traditional drugability properties. Compounds such as protacs, molecular glues, peptidometics, and macrocyclics, all of which are posing new and challenging issues for formulations. And there's tension also between sort of globalization and localization in this current geopolitical climate, and the shifting supply chains, the idea of reshoring or nearshoring strategies. So there's this that we have to contend with as well. And the regulatory landscape is evolving quickly, with new accelerated pathways opening up for pediatric formulations, orphan drugs, et cetera. And patient centricity matters now more than ever with the rising cost of medication, increasing pill burdens, complex dosing regimes, all that are really challenging patient adherence that we can address through formulations. So now let's talk about AustinPx's approach in this climate that we're in. So we're built on three pillars, starting with a sponsor-informed perspective. So we give clients direct access to our scientists and integrated project management and build strategies around real clinical trial needs because we were a former sponsor. We understand our clients' issues going beyond the science because we've been in their shoes and can advise them accordingly. Second pillar, a flexible, data-driven approach with fast execution in the face of shifting timelines. We provide transparent decision-making and platform-neutral solutions. So we let the data drive everything with full transparencies, and we remain nimble to adjust to the inevitable challenges and curveballs that come up during development. And third, we're designed for this complex landscape. So we adapt to evolving programs, we can anticipate downstream clinical and manufacturing needs, and balance speed against risk with high confidence. So in terms of our company highlight, so a quick snapshot of Austin Pharmaceutics. We have a proven management team and a talented workforce of over 40 professionals. We provide end-to-end early phase development and manufacturing expertise with specific focus on complex formulations, although we like the easy projects as well. We've got over 20 years of innovation with our KinetiSol technology, including two late phase clinical candidates of our own, for which we've gained company-wide experience that continues to help inform our partners towards the achievement of their development targets. We've partnered with more than 10 of the largest pharmaceutical companies in the world and have supported over 100 molecules to date, including some of the most challenging poorly soluble compounds in development pipelines. So a quick look at our management team. As I said, I'm Dave Miller. I'm the Chief Scientific Officer. Alongside me and Chris Bruff, who's our Chief Technology Officer, the two original co-inventors of our KinetiSol technologies. And then we have Justin Keene, who's our Senior Vice President of Operations. Elizabeth Hickman is our Chief Executive Officer. Amy Kimbrough directs our quality assurance functions. And Francesco Sherrod leads finance. So this is a seasoned team of pharmaceutical development experts who've navigated the drug development journey many times before and provides that senior leadership to our team and ultimately to our clients. So AustinPx has a real global reach. We've worked with companies across the globe. We bring I&D experience across US, Canada, Australia, India, and Thailand. Those are our own former I&Ds during our years as a sponsor company. And our footprint spans sponsor relationships across North America, South America, Europe, Asia, and the Middle East. So we've really operate on a global scale. So our quality unit expertise covers CGMP compliant testing and phase-ready validation, risk-based qualification of facilities and equipment, and our quality management system that's fit for purpose for early phase development. We're trusted by top 10 pharma companies. Our quality systems have passed multiple audits from large pharma partners. Our internal regulatory experience, as I mentioned earlier, includes 7 INDs in the US, 8 Canadian CTAs, 1 UK CTA, and 1 Thai IND. And that's independent of the regulatory support that we've provided for our numerous clients. And on the clinical side, we ourselves have sponsored 8 phase 1 and phase 2 molecules across 5 different CROs that we've partnered with and bring that experience to the table when advising our clients. In terms of our scientific expertise, it spans 3 primary areas, starting with the most complex molecules, the brick dust, the grease balls, the highly potent compounds, the chemically labile compounds, complex molecules like macrocycles and PROTACs, peptidomimetics, molecular glues. We've seen it all, and we've worked with the hardest molecules in the contemporary development pipelines. Second, we are highly focused on understanding bioavailability and ways of enhancing bioavailability through the use of either supersaturating drug delivery systems, amorphous solid dispersions, self-emulsifying drug delivery systems, cyclodextrin complexes, engineered particles. We're also quite good in terms of developing modified release dosage forms, and all are tailored. These approaches are all tailored to sort of unlock the absorption potential of these molecules and optimize PK for enhanced therapeutic outcomes and improve safety profiles. Third, we're great in terms of accelerated drug development and doing rapid assessment of the compound's properties and the primary parameters for the development program. We've got quick to clinic CTM supply in a number of different ways, which I'll expand on in a moment. We provide CGMP manufacturing through phase 2 and seamless tech transfer to CMO partners for phase 3 registration and commercialization. So from preclinical insight to clinical momentum, so AustinPx supports complex programs through integrated development and manufacturing. In preclinical development, we handle complete molecular characterization, preformulation testing, data-driven technology screening, and a wide variety of bioavailability enhancement technologies. In oral drug development and manufacturing, we produce solutions, suspensions, tablets, capsules in both immediate and modified release, all the way from early preclinical studies through phase 2 manufacturing. So in terms of our development and manufacturing capabilities, this slide shows our full development and manufacturing continuum from discovery through commercial. Early on, as I mentioned, we do complete molecular characterization, supplemented with in silico tools that allow us to establish developability assessments, and also looking at preclinical and toxicology supplies. Moving into formulations, we cover analytical methods development, complete technology screening, both for quick to clinic as well as bioavailability enhancement, and again, manufacturing of suspensions, capsules, and tablet formulations. And then from there, process development, GMP manufacturing of the final dose, all the way through phase 2, and then we can supply KinetiSol drug product intermediate at all stages. And quality is built throughout with QC testing, stability testing, storage under a fit for purpose quality management system. So we have the capability for comprehensive characterization and preformulation. We've got a complete toolkit across 4 areas. So first, looking at the key properties of the molecule. This is the fingerprint of the compound that really direct the path forward, along with the key parameters of the development program and the target product profile. So we leverage in silico tools extensively in analysis of the compound, but also looking at the physiochemical properties such as pKa, hygroscopicity, solubility profiling across aqueous, pH, biorelevant solutions, as well as organic solvents. Our biopharmaceutical capabilities, we focus on looking at the log P of the drug. In the case of supersaturating drug delivery systems, analyzing liquid-liquid phase separation, intrinsic dissolution rates, solubility in biorelevant media, as well as trans membrane flux, and always CACO2 permeability. With solid state characterization, again, a full toolkit to evaluate crystallinity, polymorphism, thermal analysis, particle size, crystal habit, powder rheology, and in terms of stability, looking at pH stability, thermal acid base, oxidative degradation, photodegradation, and dynamic vapor sorption. So all this is designed, all of these tools, to inform our formulation strategy, manage risk, and support scalable development. When it comes to data-driven development, our workflow moves quickly from pre-formulation, supplemented with in silico tools, to arrive at a developability assessment, enabling a decision towards either bioavailability enhancement, formulation enablement, or quick-to-clinic approaches. So if enablement is needed, we have the widest, we screen the widest range of enabling technologies, from particle size reduction to lipid and self-emulsifying systems, as well as amorphous solid dispersions, which encompasses both spray drying, hot melt extrusion, KinetiSol, and anti-solvent precipitation methods. So we've got under one roof the widest range of ASD technologies for which we can screen early and make the best decision based on the data. If bioavailability enhancement isn't required, we move straight into conventional sort of speed to clinic development with approaches such as API in a capsule, powder in capsule, powder in bottle, or we can go beyond to dry granulation, wet granulation, encapsulation, tableting, and film coating. So either path feeds into our biopharmaceutical analysis tools, and as well as accelerated stability testing, which allows us to generate clinical prototypes, ticking all of the boxes in terms of performance, stability, manufacturability, and ultimately a rapid transition over to clinical trial material manufacturing. So when it comes to fast and fit for purpose prototypes, our speed to clinic strategies take a few different forms. As I mentioned, we can go as simple as API in a capsule using our fill to weight system. It can be in a capsule, a bottle, or a vial. The dose ranges under 1 milligram up to 5 grams with less than 2% RSD, handling even very challenging powder fills, electrostatic powders, cohesive powders. Still work well within the fill to weight system and accelerate entry into clinic with a very simple dosage form. Beyond that, we can do material sparing tablet development with early identification of compression risks, powder flow risks, leading towards data-driven optimization and using compression simulators to simulate rotary press and flag any issues that may affect scale up and resolve those early. We can do rapid capsule prototyping across blends, granules, multi-particulates, screened across a variety of capsule sizes using our capsules program. And for liquids, we commonly manufacture solutions, suspensions, syrups when needed. We handle the vehicle design as well as the preservative system design. And inherent in these is particle size control, stability, and redispersibility optimization. Good. Next slide. So when we're talking about efficient commercial ready formulation, our goal is to deliver your best formulation from day one. So we have extensive capabilities for dry granulation, including roller compaction. On a smaller scale, we can do slugging in a simulated roller compaction scenario. Material sparing approaches for dry granulation design are really critical early on, and then it branches into seamless scale up towards clinical trial material manufacturing. In terms of wet granulation, we have both high and low shear methods, fluid bed drying with robust granule formulation and property control. For tablet and capsule development, we can do immediate, modified, and extended release. We're also quite proficient in developing mini tablets and fixed dose combinations. And once again, clinical trial material manufacturing up through phase two, or maximum batch size of about 150 kilograms out of our facility. And in terms of coating, we do cosmetic coating quite often, as well as functional coating through pan and fluid bed systems for, just either cosmetics or taste masking, gastric protection, or providing delayed or extended release. Not too far. And then go too far. There we go. For bioavailability enhancement, we offer a complete suite of bioavailability enhancement tools. First, looking at micronization to particle size reduction to achieve dissolution gains down to particle sizes as low as about 1 micron. Also looking at lipids and self-emulsifying systems. We're looking at vehicle solubility, combination formulation solubility, as well as stability, dispersibility, precipitation evaluation, digestion, and dissolution performance. Our bread and butter is amorphous solid dispersions. So we have the widest technology screen of any company in the industry, encompassing our own KinetiSol technology with spray drying, melt extrusion, and anti-solvent precipitation, with full formulation stability and biopharmaceutical analysis. We also have a unique capability for evaluating cyclodextrins through our KinetiSol technology. So KinetiSol is able to form solid state inclusion complexes through frictional processing in a scalable manner that gives complete complexation efficiency at the right stoichiometric balance. And those often provide dramatic improvements in solubility and flux, and high levels of inclusion complex stability. So I mentioned we have the widest range of technologies for amorphous solid dispersion evaluation. So this slide provides a deeper dive on our capabilities. So first, we have our proprietary KinetiSol technology. It's a non-solvent heat-sparing approach to developing ASDs with the widest formulation design space, which encompasses more molecules and more excipients and mixed matrix optimization approaches, which we can prototype very rapidly at our formulator scale KinetiSol unit. Next, we do spray drying with the ProSet 4 matrix system. So this is lab scale spray drying for rapid screening and prototyping, and also small GMP runs. We can develop STD prototypes, like I said, through a full formulation screening protocol and feed those into our in-depth characterization approaches for solid state chemical and biopharmaceutical analysis. We do early stage hot melt extrusion evaluations using the melt prep vacuum compression molding tool. This allows us to assess HME on a very API sparing basis, milligram quantities that allows us to screen a variety of different polymers, looking at thermal stability as well as performance of the melt extruded dosage forms. Also, solvent anti-solvent precipitation is something that we do occasionally when all else fails, and it's primarily focused on anionic polymer screening and precipitation conditions that provide ASDs with significantly improved solubility and ideal particle properties for downstream processing. And screening each of these platforms includes in-depth biopharmaceutical analysis in vitro and in vivo PK, as well as stability, both chemical and physical, as well as looking at a comprehensive set of dissolution approaches depending on what's needed for the molecule and the dosage form, from biorelevant to intrinsic dissolution to even non-sync methods where appropriate, and multi-phase methods. So our characterization of enabled formulations is very comprehensive. We have the complete analytical toolkit. For solid state analysis, we have powder X-ray diffraction, modulated DSC, microscopy, including polarizing light microscopy and scanning electron microscopy, as well as particle size distribution. For chemical analysis, HPLC, UPLC, LCMS to fully characterize the dispersions in terms of assay and related substances. For stability, we use a variety of different accelerated conditions depending on the needs of the program and the formulation, and then evaluate through chemical and solid state analysis as well as biorelevant in vitro testing to ensure performance on storage. We have a very comprehensive toolkit of biorelevant dissolution approaches as well as transmembrane flux. We have several pion micro and macro flux systems that we leverage regularly to provide within one experiment both a dissolution result and a transmembrane flux result. And we find comparing those to in vivo PK, we can often arrive at an in vivo in vitro relationship very quickly. For lipid systems, providing dispersion digestion analysis. We also have the capabilities to look at the speciation and determine how speciation is driving absorption. And of course, in silico modeling to supplement all of the in vivo and in vitro testing. So I've mentioned our unique KinetiSol technology. Let's talk a little bit more about that and how it's differentiated. So it's a very scalable technology for amorphous solid dispersions. In fact, we developed KinetiSol starting at an industrial scale based on a recycling technology. So it's a technology that had initiated at a very large scale and then scaled down. So it's quite the opposite story of most new technologies in pharma. So it's well vetted in terms of its scalability and ability to produce product at volume. So it's a very simplified processing system with very consistent operating principles that enable predictable scale up from bench scale, gram quantities, all the way to kiloton quantities. And it's a highly sustainable technology, a very small footprint. We use no organic solvents, no purified nitrogen isn't required. There's no secondary drying of the ASD material because there's non-solvent. And as we go from research to manufacturing, there's a single step towards the manufacturing scale. And once at the manufacturing scale, the equipment remains constant. It's the duration of processing that changes. So there's no scale up risk beyond phase one. So as the program progresses into later stage clinical development, it remains on the same equipment, and it's merely the duration of processing that is extended. So your material properties and your manufacturing properties remain the same from phase one all the way to commercialization. And KinetiSol really delivers on performance. And I mentioned before, it has the widest formulation design space of any ASD technology, where we can process any API independent of melting temperature or thermal lability because of the way the thermal process works. It operates on the order of seconds and not minutes, and the material only sees elevated temperatures for a few seconds. So that's how we're able to process APIs spanning the range of melting temperatures and thermal sensitivities. And beyond that, the excipient space that we can evaluate is vast. We're not limited by solvent solubility of excipients or thermal properties like viscosity, high TG, lack of thermoplasticity, or thermal sensitivity of excipients. Those don't encumber our formulation space. So we're able to, and often do, create multi-component matrices to simultaneously solve the biopharmaceutical needs as well as chemical physical stability and manufacturability, leveraging a very wide toolkit and using combinations of materials to solve those problems. And in terms of commercial manufacturing operations with KinetiSol, we offer multiple pathways to commercial scale manufacturing. We do full formulation process development here at Austin Pharmaceutics, and we manage the tech transfer to a preferred partner, which can be either direct contact with our strategic partner in Thermo Fisher Scientific or subcontracting through another partner such as Microsize. We can also place equipment at a client's preferred CMO via a lease agreement, or placed directly at the client's own commercial site, again, through a lease agreement. And this flexibility means that clients can choose the commercial path that best fits their long-term strategy. I mentioned Thermo Fisher a moment ago, so let's take a minute to talk about expanded commercial access through our partnership with Thermo. So, as a major recent milestone for us is our agreement with Thermo, and this expands our commercial manufacturing access and strengthens the pathway for KinetiSol-enabled programs from development through commercializations. So, it gives our clients broader options for commercialization. It expands our KinetiSol ecosystem and provides continued access to AustinPx-led KinetiSol expertise. You have two options in terms of developing KinetiSol formulations and manufacturing. So again, it's scalable, sustainable, and improved performance. That's what KinetiSol offers, and it's now backed by Thermo to shore up its commercial reach. So let's shift a bit to talk about speed to clinic cGMP manufacturing. So this is what it looks like, what it actually looks like once we have the API in hand. So from receipt of API, we do rapid pre-formulation testing, formulation screening, drug product development, scale up, method validation. These all run largely in parallel with any needed animal studies. And then from the time we receive GMP API, we're writing batch records, executing the manufacturing, testing, release, generation of a COA or COC, packaging, labeling. And the bottom line is we can move from GMP API receipt to phase one CTM shipment in just 6 to 10 weeks. Our systems are set up to provide this type of speed and flexibility. And then in terms of potent compound handling, we have a fully integrated approach. Handling potent and hazardous compounds safely is really critical. Many compounds are highly potent these days, and we take a fully integrated approach to handling these compounds. So we use the AffAgility 5 category band based on occupational exposure limit, ranging from fully permitted to full containment, and analyze these on a case-by-case basis. So we collaborate with toxicology and occupational health experts to assess the molecule's characteristics and toxicology data and tailor our approach to each molecule and project needs. Additional support in terms of safety includes gap analysis, waste management, employee training, and EH&S expertise. So now let's look at a client case study focusing on speed to clinic, and I'll walk through this real example. The challenge was it was an early stage biotech company with limited API that needed rapid phase one entry, and a CDMO that could support both their immediate clinical needs as well as the future fixed dose development. So needed to get into clinic quickly with a fit for purpose dosage form, but with an eye towards ultimately developing the long-term formulation. So our solution was, once again, starting with complete molecular characterization, looking at pre-formulation and compatibility work that guided near and long-term dosage form design. We selected an API and a capsule feasibility approach for rapid phase one entry, optimized the analytical methods, and fully integrated program management to help accelerate all of those moving parts towards delivery of the phase one supply. The outcome here was we were able to provide GMP capsules that were delivered on time for a global phase one study, advancing from initiation to clinical trial material in just 4 months, with early data that not just inform phase one supply, but also support a path to a scalable, commercializable tablet. Next client case study is pediatric dosage form development. This is a mini tablet formulation approach. And this case study covers the clofazimine pediatric solid or oral dosage form development. We did a webinar earlier this year with our partner that I encourage you to find on our website for full information. So the challenge here was the API was micronized, very low bulk density. It was a highly compactable API, very poor flow. There was capping risks, and there was a need for a mini tablet dosage form with 50% drug load per mini tablet. And the mini tablet was needed because suspension dosing um, wasn't sustainable for this particular drug for chronic pediatric use. So our approach, we moved from the transferred direct compression approach, which didn't work, through evaluating dry granulation, high shear wet granulation, ultimately had to modify the formulation and substitute excipients to improve flow and compression properties to achieve the dose strength and the drug release. And we were able to do this leveraging our Style 1 Nano compaction simulator and our FT4 powder rheometer that allowed us to flag our issues early, pivot with the technology and the formulation. And the outcome was we arrived at optimized pediatric mini tablets with the target 50% drug load, balancing compactability, mechanical strength, and dissolution, and demonstrated scalable CTM manufacturing pathway. What one element of this development that was added complexity was that these tablets needed to be film coated for taste masking and masking of this very red drug substance from teeth staining. So the mini tablets had to be developed to withstand fluid bed coating, which is how we coated the mini tablets with the taste masking coat. So we were able to do that and provide clinical trial material manufacturing of a very complex drug product in a very short amount of time. Finally, last case study here involves late stage reformulation of a suboptimal spray dry dispersion. In this case, we pivoted the client from spray drying to KinetiSol based on the specific challenges of the molecule. So the client came to us with the challenge was they had through phase 1, phase 2 spray dry dispersion tablet with suboptimal bioavailability that was leading to high pill burden, 5 tablets per administration, and significant manufacturing complexity from a process that involved low solids load in the spray dried feed from a solvent that was very difficult to handle and limits the large scale spray drying capabilities to CMOs who could handle that particular solvent. So our outcomes with KinetiSol, we applied the KinetiSol technology. We optimized the formulation. That our optimum prototype went into a primate study. It showed 5-fold increase in AUC, reducing the projected pill burden from 5 to a single tablet. And we were able to reformulate and provide these prototypes within 4 weeks from supply of R&D API. And then once that primate study came back and the client was eager to move forward into a human relative bioavailability study, we were able to do that, manufacture clinical trial material in just 4 months. And not only did we provide substantial formulation improvement, we also substantially reduced the manufacturing risk by eliminating the need for solvents and the low solids load that was affecting volumetric efficiency that resulted in a 67% reduction in manufacturing time. And as I mentioned before, much simplified scale up with KinetiSol over spray drying. So I mentioned 5-fold increase in primates and then supply to a human relative BA study, and it confirmed meaningful improved exposure with the KinetiSol tablet compared to the original SDD tablet. And now we're engaged in manufacturing the phase 3 supply for this program. So finally, why should you partner with us? Again, just to reiterate, we have a depth of expertise in bioavailability enhancement. It's been a large part of our focus over the history of the company. So we have an extensive bioavailability enhancement toolbox. We screen all the technologies from ASDs to lipids to particle size reduction cyclodextrin, and we can screen these and let the data decide which approach best fits the molecule and the development strategy. And as an expert in bioavailability enhancement, we have a very deep biopharmaceutical analysis capabilities from looking at solubility in biorelevant media to transmembrane permeability, of managing animal PK studies and feeding that back into our in vitro and in silico evaluation to help arrive at a full understanding of the dissolution absorption process in vivo. And these are all executed or managed in-house, ensuring that every formulation that we work on is optimized for performance. And then finally, we always develop these formulations with scale in mind. We from day one, as we're developing formulations, even at a small scale, flagging issues that might limit larger scale manufacturing and either resolving those or pivoting to alternative technologies, and and generating data and processes that hold up as the molecule progresses through clinical development. So with that, I would like to thank you for your time today. Thank you for listening to the complete comprehensive capabilities of AustinPx, and we love the opportunity to discuss your specific program and how we can help. You can reach us either at info@austinpx.com or by phone at our main line there listed on the screen. And free to visit our Georgetown facility for a tour at any time. And you can follow us on LinkedIn using the QR code on the screen, and we look forward to connecting with you in the future. Once again, thank you for your time.